Really excited to be able to talk with you all about some of my work. And so today I'm going to be talking pretty broadly about some projects I've been doing over the last couple of years, which really all focus on kind of developing kind of advanced modeling techniques, advanced spatial modeling techniques that can be applicable for forest and wildlife management. And so my plan for the talk today is to kind of give a broad overview, kind of a broad description of the types of models I work with, go a little bit into some of the software that I use to do that, and then we'll wrap up with a couple of applications of the work. Okay, and so I'm going to start off with what's seemingly a very simple question, but just kind of making sure we're all on the same page here. So what are our spatial data? Because these are the types of data that I work with, and I'm sure many of you all work with to understand complex ecological processes across space. And really, spatial data is any type of data that relates to a specific geographical area or some location. So it can be associated with specific latitude and longitude coordinates. It can be some sort of aerial unit, but associated with some sort of location in space. And these are the data that form the foundation of spatial statistics, and kind of a key founding principle of spatial statistics is what's called the first law of geography, which is this unbelievably simple law, but also really powerful and kind of developing the motivation for a lot of statistical tools. And the law states everything is related to everything else, but near things are more related than distant things. Okay. It's an unbelievably simple concept. I don't think there's many laws that are as simple as this law, but really this information can be applied to a lot of the types of analyses that we may do with modern ecological and natural resources data. And so in particular, in this age of large-scale data collection and big data, we're seeing these massive amounts of spatial data and also spatiotemporal data as well. Okay, and so here I have two data sets I use pretty prominently throughout my work, data from the North American Breeding Bird Survey on the left, as well as data from the U.S. Forest Service on the right. And these are two data sets that provide us with massive amounts of information to understand bird population dynamics over space and time, as well as just tons of information on forest resources available across the US and how those might change into the future under different management regimes and under different scenarios of climate change. And so, as you might know, these data are large nationally funded programs, and they take a lot of effort, a lot of time to generate these types of data, these large spatial data, these large spatiotemporal data. But there's also kind of these newer ways that we're getting increasing amounts of spatial and spatiotemporal data that are really transforming the types of questions that we can answer across very large spatial scales. Okay. And so this includes things like citizen science programs. Individuals in the public will go out, record different observations that are then fed into databases, and we can analyze in all sorts of different ways. And it also includes different types of kind of autonomous approaches to monitoring natural resources. So things like acoustic recording units, camera traps, and a whole suite of different types of remote sensing products that can really provide us with just tons and tons of info to address not only fine scale and local scale management questions, but really broad scale management and conservation questions that drive a lot of the types of work that we do. And as you might imagine, these data are collected, they're different approaches to collecting data are going to lead to a whole suite of biases in these types of data, depending on the way that they're collected, right? And so we know that with ecological data comes a whole suite of observational and sampling biases, okay? So things like false negatives, false positives that commonly occur in ecological data need to account for those things when we go to analyze them to get the most robust inference and ultimately recommendations that may come from these analyses. And then also, with this increasing size of data, with these spatial data that we have, fitting these models, doing these analyses can take very large amounts of time. So we need to think about how we can improve the models to address this ever increasing amount of data that we have available to us. And so kind of in addition to these complexities with the data, with the size of the data, we also have just working with complex ecological processes, right? So we know that things like population dynamics are correlated across space and time. So we should be accounting for that in our analyses. When working across really large spatial ranges, we often encounter the concept of non-stationarity or context dependence. Okay, so this concept that the relationships between a species and its environment may be different in one part of its range compared to another. And then, of course, we know that species are not independent of each other. We should be accounting for these species if we're working with multiple species in our analysis, and using that to improve the information that we can get from these models. Okay. And so really, this slide here kind of sums up most of my work and kind of the applications of my work, where I'm kind of developing different modeling approaches that can be used to address these different complexities. And by doing that, provide us with the ability to inform conservation management questions across different scales. Okay. So again, just kind of quickly going over what I'm going to talk about today, going to really kind of give a little bit of insight in terms of the spatial statistics methods I use. So go a little bit underneath the hood in terms of the types of models I use. Quickly discuss how using those methods, developing those methods also requires the development of software that managers and practitioners can then use, and then get into some applications of that work where we can see how these approaches can be used to inform conservation and management. Okay, so first thinking about, let's just set up the scene here. Let's think about the data that we might be working with, and potentially that you may work with as well. A lot of my work is motivated by estimating distributions, abundance, and growth or trends over time, which are all really kind of some of the most basic management and conservation tasks that we can do. Okay. And so what the data I'm talking about are going to look like kind of on the right over here, where you can imagine that at each of these locations, you have information on the presence or the absence of species you're working with, or some measure of abundance, which can be counts or some measure of biomass or whatever you might be interested in. And importantly, you have the spatial coordinates for each of those locations. Okay, so these are the geo-referenced, point-referenced spatial data. And the goals for this type of analysis could either be prediction or inference, or both. Okay, so different types of objectives for different sorts of questions. And of course, we can think about the most basic kind of framework that we can use to analyze these data. So thinking about our classic GLM, where we're going to have our response variable y. Here I'm denoting that y with this bold S to indicate that y is at different sets of spatial locations. Where this bold S corresponds to the coordinates for each location I that we have. Okay, we're going to have a total of n different locations. And then with a classic GLM, we can include different sorts of covariates that are related to our response variable with some regression coefficients, and have some sort of dispersion parameter associated with that. Okay, so a classical model used throughout ecology. And then just taking a look at what this would look like in equation format, right? We have some distribution that we link this mean through a link function and relating those covariates back to whatever response we're interested in, okay? It's a really classic model used for a whole suite of purposes. And the key assumption in this model is that these y's are conditionally independent of each other, okay? And so when we think back to that first law of geography, right, that kind of implies that these spatial data are routinely violating this independence assumption, okay? And so what does that sort of concept of conditional independence mean? It means that after you account for the covariates in the model, are your residuals or is there additional spatial correlation that exists in the data that you're working with, okay? And again, kind of these spatial data routinely violate this assumption. So we need kind of new methods to kind of address this, okay? And the classic kind of spatial statistics approach to doing that is kind of a spatial generalized linear model. Okay, so we're adding in kind of an additional layer here to account for the spatial autocorrelation that may exist because things closer together might be similar, more similar to each other than things that are farther apart. Okay. And so what do we do? Well, this is a very classical framework. We again have our response variable, and the only difference we're doing is we're adding in this W here, which is a spatial random effect, which is essentially just going to adjust the mean up or down at each location, depending on what the data are suggesting, okay? And with these spatial effects, these spatial random effects are governed by this k theta, which is a big spatial covariance matrix, okay, that essentially is going to kind of indicate how similar two sites are in terms of their residual spatial correlation, okay? And if we just kind of take a look at this k theta, what's going on here, we can imagine where we have 5 different sites in our data set, okay? And for this covariance matrix is going to be this large matrix where there's a value at each cell, and each cell indicates the covariance between 2 different sites, okay? So this K51, how correlated or what's the covariance between site 5 and site 1, okay? And so that's what's used, that's what drives these models, allows us to estimate these spatial random effects. And this is kind of a classic Gaussian process type spatial generalized linear model, okay? Where the only differences we do is we add in the spatial random effect here, and then that whole vector of spatial random effects comes from a multivariate normal distribution, okay? And so thinking about this a little bit more and why we may need to kind of extend it, remember that this k theta is a big n by n spatial covariance matrix, okay? And this approach is really useful because it is flexible in terms of the kind of spatial correlation that it can estimate, okay? So if we take a look a little bit in terms of what governs the correlation between 2 sites, there's really kind of 2 things going on here. There's one parameter that controls kind of how much variation there is in that random effect over time, okay? So just like any sort of random effect, there's a variance associated with it that controls that variation. And we can see how that impacts what our spatial correlation might look like as we change that parameter from small values to large values. We can see that there's much larger magnitude differences across the different sites in this data set. And then what we have is this phi here, which is kind of a spatial decay parameter, which controls kind of how far across space the correlation between 2 sites goes. Okay, so you can see here where phi is large, the correlation only goes across very small distances, but as you decrease that phi, move down this graph, you can see that the correlation extends across much farther spatial areas, okay? And so this approach is great. And so this spatial Gaussian process approach, well developed in the spatial statistics literature, has been for 40 years or so, widely applied. But where we start to run into problems is where we have extremely large spatial data, okay? And so this is what's called the big N problem in spatial statistics. And so I have a little bit of details here about what we need to compute when we estimate this model, okay? So fitting this type of model requires doing this computation on the right. And all that you need to know is that these 2 operations here, taking the determinant of this big matrix and the inverse of this matrix, those are extremely intensive operations, okay? And so what happens is the complexity of this model is going to increase with the cubic order of spatial locations. So as that number of spatial locations you have increases, the computational complexity of this model is going to get extremely infeasible, very difficult to work with, all right? And so this means that this model, even for pretty moderate sized data sets, so 200 locations or so, can become very, very difficult to work with, okay? And so this is kind of where we need to start thinking about other approaches, because if we do some sort of simple simulation, so here I have about 1,600 locations for a specific type of model called the spatial occupancy model. If I estimate this using a Bayesian framework, what we can see is on the left here, the true spatial effects I'm trying to estimate. And then on the right, our estimate that we get from the model. And we can see that the estimate does a pretty good job, right? They look pretty similar. It's getting pretty close to the truth. But we can see that it takes about 7 hours to run, right? So that is not going to be a really feasible model, especially as the size of our data just keeps growing and growing. Okay, so thinking about some approaches we can use to get around to that, the approach that I use in my work is what's called the nearest neighbor Gaussian process approach. Okay, and the concept behind it is relatively intuitive. You can imagine this big covariance matrix, right, where you have points at where you have cells, and in each cell there's going to be a non-zero value. The whole concept of the NNGP is that you're going to strategically choose some of those cells to be zero, and then you don't have to work with them in a very computationally expensive way, okay? And so the whole theory behind how this works is if you have this multivariate normal distribution here, what you can do is assign some order to the spatial locations in your data set, such that you have a first location, you have a second location, a third, and so on and so forth. And then you can rewrite that as kind of a product of a bunch of probability densities where they're all sort of conditioned on different locations. Okay, so for your first location, you have probability density of just that location. For the second one, you multiply that by the first, and notice that the second is conditional on the first location. For the third, conditional on the first two, and then so on and so forth until we get to the final nth value. It's conditional on all the previous n-1 values, okay? And so the computational problems arise in these models for these types of densities on the right here, where if your n is in the thousands or the hundreds of thousands, this conditioning set is extremely, extremely large, and that leads to the difficult computations, okay? So the whole concept of what this NNGP is going to do, essentially going to set some maximum number of values on the right-hand side of this distribution, and choose those values in a way that results in a minimal loss of information, okay? Okay, and thinking about this from a graphical perspective, because that can be a little bit easier to think about, we can draw all this in terms of a directed acyclic graph, okay? So what each of these nodes represents is a spatial location, and then each of the arrows indicates kind of conditional dependence on that location, okay? And so what we have here is this first location is mapped to this first node. Notice there's no arrows going into it. The second location is right here, and we have it conditional on the first, the third, so on and so forth throughout this graph, okay? All the NNGP is doing is it's saying, let's set some upper bound on the number of arrows that could be pointed into each node, and then strategically choose which ones we want to keep. And then we're going to drastically reduce the number of arrows that we have in this graph, okay? And so here, I'm restricting the number of arrows to each node as 3. And so we kind of choose which ones we want to chop out in these conditioning sets, okay? And again, the whole concept of this is this neighbor set or this conditioning set, you're going to restrict that to some value, to some maximum size of arrows to point into an individual node, okay? And usually the best way to do that is, as I'll talk about in a second, is just to choose those locations based on the distance of the neighbor locations to each other, okay? Okay, and then if we take a look at what this does, all this does here is this is a covariance matrix for 1,000 locations. Okay, with the full Gaussian process that I talked about before, this entire graph would be black. Those black points represent the points that are non-zero. With this NNGP approach, we essentially put in a ton of zeros into this covariance matrix. And so the only non-zero values we have to estimate are these black points, which is a much easier approach to do from a computational perspective, okay? And then if we think about what this does, the beauty of these NNGPs is that the model is exactly the same with just only one difference. The only difference is there's just a change in that covariance matrix that we use, okay? So that covariance matrix is slightly different. It depends on that formulation that I just described. Okay, and what this does is it allows us to use this much more efficient approach in any way that we could use the standard Gaussian process approach to work with spatial data, okay? And throughout, I use a Bayesian framework in my work, and most of these are estimated using Markov chain Monte Carlo as well. Okay, and if we think about the trickiest part of fitting these models is determining which of those arrows to chop out, okay? And so the kind of term that we use for determining which are the arrows pointing into each node is called the neighbor set. And it all relies on kind of some basic spatial statistics concepts where essentially the correlation between two sites is going to decrease as sites move farther away from each other, right? So that first law of geography. And this all relates to this concept of what's called a screening effect, where most of the information to estimate a spatial covariance function is contained within the first few the sites that are closest to any given location, okay? So in other words, we're going to choose the neighbors based on the sites that are closest to any individual site, okay? So the process to doing this, figuring out the neighbors, first thing you need to do is give some order to the spatial locations, okay? So that means choosing some way to order locations across two-dimensional space, which may not be the most intuitive thing, but there's been some work to show that that ordering is not necessarily all that important. So we'll often just order along one axis. And then once you do that ordering, figure out the nearest neighbors to your site that you're working with based on some simple distance metrics, so linear distance or Euclidean distance, okay? And so thinking about what that's going to look like, okay, if we have our locations across two-dimensional space here, if this is the current site I'm working with and I want to figure out the neighbors I need for my NNGP approximation, what I'm doing here is I'm going to order the sites based on their location along the x-axis, okay? And so what that means is that all the potential neighbors for this site are only those that are on the left-hand side of this graph. In this case, I'm going to choose 5 nearest neighbors. So I'm going to take the 5 sites that have the smallest Euclidean distance to that site, okay? Same thing with the approach on the right, all right? So all the neighbors have the potential values to the left of this line, and just calculate the 5 sites with the smallest straight line distance to those sites, okay? And again, this kind of shows the ordering restraint here. So we have, notice these sites are a bit closer to it, but because they're on the right-hand side of the line, they are not included as neighbors in the model, okay? And again, kind of the other thing you have to think about as well, how many neighbors do I want to choose in this model? Okay, well, to do that, what you would do is kind of look at some work that's been done testing different simulations. And what this graph here shows is that as you increase the number of neighbors to about 15, the approximation for the NNGP is essentially identical to what we are trying to estimate, that full Gaussian process, okay? And it does all this in a much more computationally efficient way, okay? And so with just 15 neighbors, we can get a great approximation of the model we want to look at. Okay, so all that said, let's take a look at the results that gives, that this approach gives us. If we take a look back at our simulation here, we add in this third panel, look at what this NNGP approach is going to do. We see that we get essentially the same plot between the full GP and the NNGP, but now notice there's a 7-minute runtime for this NNGP compared to 424 minutes, okay? So substantial improvement in runtime. Okay, so that's kind of the NNGP side of things, but with these sort of statistical models, okay, you develop these statistical models and you want them to be used for different applications. But of course, in order to do that, we need to develop software along the way that allows us to to take these models, package them up in a user-friendly system, and allow those to then be used by practitioners and managers to apply these tools to different sorts of questions, okay? And so what I've done over the last couple of years, developed a couple of R packages that take this approach and package it up into R packages that could be used to apply these models to different systems applicable to ecological research, okay? And so these packages are SP occupancy and SP abundance. So SP occupancy is used to fit a specific type of species distribution model. It's called an occupancy model, where those occupancy models are a type of model that accounts for false negatives in a lot of collection of wildlife data. If you're wondering what's up with the logo on the right, well, you got to do a little bit of kind of tricky thinking here, but if you say spatial occupancy, that's kind of where the SP occupancy came from. If you say it as one word, SPA occupancy kind of sounds like Spock, right? So that's kind of where that came from. I won't tell you how long it took me to come up with that. But anyways, that's the first package. Then the second package is SP abundance, which is similar. Fits a variety of different types of models to estimate abundance, density, biomass, using these types of spatial modeling techniques, okay? And both of these packages kind of designed in a way to be both efficient as well as user-friendly in the sense of not requiring users to have to write a whole bunch of code themselves in order to fit these models. Okay, so let's kind of think about now how these can be applied to different scenarios, all right? So the first kind of application I'm going to talk about is kind of a long-running project doing with colleagues at National Audubon Society as well as Michigan State, where we're looking at estimating spatially varying species environment relationships in grassland bird communities, okay? And so first kind of thinking about the kind of motivation of this work, on the right is just an example distribution of Carolina chickadee during breeding season. And, you know, one of the most fundamental questions in ecology is just understanding what drives where species occur over space and time, right? And there's really increasing interest in understanding how global change is influencing distributions. Are those distributions going to shift northward? Are they going to contract? Are they going to shift in some other ways as a result of global change? And so a key assumption in a lot of modeling frameworks is that the effect of global change is going to be consistent across a species range. But this is not necessarily the case, okay? So we might more so expect the effect of global change to be different across the range of a species, or that effect might be non-stationary or dependent on the context, okay? And so as an example, you can imagine that the effect of increasing temperatures, so more extreme temperatures might be more, have a higher impact in the southern part of the species range compared to the northern part of its range, okay? And so getting a model that's able to accurately estimate these complex spatially varying relationships between species distribution and global change is kind of a necessity in terms of allowing us to be able to better understand what's driving species distributions and its relationships with the environment over time. Okay. And so what we sought to do in this work is kind of extend that basic, that broad spatial modeling framework that I talked about to allow us to estimate these sorts of non-stationary species environment relationships. Okay, so allowing the relationships between a species and its environment to vary across space. Okay, in particular, this was all kind of motivated by really trying to understand global change impacts on grassland birds or birds more broadly across the US. And here focusing on understanding the relationship between maximum temperature and the distributions of 21 grassland birds. Okay. So thinking a little bit about what this model is going to look like. This model is called the spatially varying coefficient model, spatially coefficient occupancy model. And so what we're going to do, we're going to kind of break it up into these different sections. And so first, we're going to model the ecological process, so the distribution, the occupancy of our species, where we have a 1 or a 0 for each species J at each spatial location I. What we're going to do is we're going to model that in a GLM type framework where we look at the effects of different environmental variables, okay? And we're also going to account for spatial autocorrelation. We're going to include spatial random effect in our model, just like what we talked about. In particular, what you can notice here is that the effects of these different environmental variables, we're going to allow them to vary not only by species, so that's what this J is, but we're also going to allow them to vary across each location in our data set, okay? So we know that species have different relationships with the environment, but now we're allowing also those relationships between the environment and an individual species to vary across different locations. Okay, then what we're going to do when we collect ecological data is often we have some biases in it. And so in this occupancy model framework, we collect multiple surveys of data at each location, and in order to account for false negatives that occur in this data collection process, okay? And so additional component to this model is accounting for those observational biases. Okay, and then what we do, because we're working with multiple species, we're going to link these individual species together at a community level, where this is going to take a couple of different forms, where first we're going to be estimating these species-specific effects as random effects, so linking them in a community level distribution. But also what we're going to do is we're going to account for correlations between the species, both in terms of their residuals as well as the effects of these different environmental variables, okay? And so building this model, let's think about how well this model does, okay? So first we're gonna test it out under some different situations. And what I have shown here are 6 different potential species environment relationships, okay? Where blue indicates a positive effect and red is a negative effect, white being no effect. And these vary from extremely simple, so just a constant relationship across the whole space, to some pretty complex spatially varying patterns on the right, okay? And if we look to see how well our model does, underneath this panel, I'm going to show the estimates that come from our spatially varying coefficient models. And what we can see is the model does a really good job at approximating these complex patterns and these complex relationships, okay? So we can see it does a good job of capturing these spatially varying patterns on the right. But then when the relationship is more simple, we also see the model kind of simplifying down and revealing that more simple pattern, okay? Okay, so that's great. Built this model, looks good. Let's apply it to a case study. Okay, so again, here we are working with grassland bird communities. And these data come from the North American Breeding Bird Survey, specifically working with 21 grassland birds across about 2,500 BDS routes, okay? And again, our whole objective here is to understand if the effect of maximum temperature varies spatially across the species ranges, okay? Because understanding if it does, that could provide us with some insight on if increases in temperature would lead to shifts in a species range northward or potentially towards higher elevational locations. Okay, so let's fit the model to these data, take a look at the results. And here what I have shown is kind of a summary of this whole complex analysis that showcases how these effects vary across species and across space. Okay, so kind of walk through this figure here, where what we have on the x-axis are individual species, and the y-axis represents the proportion of the spatial locations where the effect was either strongly positive, as shown in blue, or strongly negative, as shown in red, okay? And so what we can see is that where if we move across the species, we see different colors across the species. That's not surprising. Different species are going to have different relationships with temperature, right? But then what we can see is that within a given species, that many of these species have both positive relationships as well as negative relationships with maximum temperature, dependent upon where we are in the species range, okay? So we're seeing evidence for a lot of spatial variation in these sorts of relationships. Okay, then what we can do is we can kind of take one of these bars, you can imagine for each of these species, and then make a map of these effects across the range of the species and take a look at what that might suggest to us about how these distributions may be shifting across, or how its maximum temperature may be influencing these distributions, okay? And so what I have shown here are the effects of maximum temperature on two different species, Vesper sparrow and ferruginous hawk, across their ranges in the continental US. And what we can see is start to use these maps to generate insights on what might happen to these distributions in the future. Okay, so for Vesper sparrow, you can see strong negative relationships with maximum temperature in the southern portion of its range, as well as in the Great Plains region. And then some other portions of its distribution, you can see more positive relationships, right? So potentially indicating with increasing temperatures, species may shift towards more northerly latitudes, okay? And you can also imagine doing this for multiple different types of environmental variables, and then using that to identify which of the different covariates in your model might be most important in driving future changes in the distribution of that species. Okay, so some of our ongoing work with these analyses is to extend this to also look at changes in relationships with land cover change, also with actual changes in climate over time. Use that to identify locations in a species distribution where climate versus land use change may be having more prominent effects. Okay. Okay, so that is kind of the first application of this work. Let's kind of move on to kind of a second application, think about how we can apply these models to a different context. In this specific work, just collaboration with Forest Service, as well as some colleagues in Michigan State and UMass Amherst, we're looking to build some models to estimate species-specific forest biomass in small areas across very large spatial regions. Okay, so in particular here, focusing on the southeastern US. Okay, and if we kind of shift gears, think about kind of the motivation behind this work. All right, so estimating forest biomass and carbon are becoming increasingly mandated by different reporting requirements, both at an international level for different sorts of greenhouse gas reporting, as well as in national and state level policies that require information on forest resources as well as greenhouse gas emissions. Okay. And in a lot of these policies and in these mandates, for example, from the IPCC, there's actually specific language that refers to a desire to kind of have unbiased statistical estimates, as well as having statistical models that can give us the smallest uncertainties as possible in our estimates of biomass and carbon and other resources that impact greenhouse gas emissions. Okay. So as a statistician, I'm extremely excited whenever I see someone actually cares about seeing the ability to reduce uncertainty. You know, I get kind of light bulbs in my brain when that happens. So I get pretty excited seeing that there's a desire for this to actually happen. And so what there's a lot of interest in doing is developing improved models that can really help reduce this uncertainty to improve these sorts of estimates. Okay. And in particular, not only kind of biomass and carbon as a whole, but also thinking about how we can improve species-specific estimates, because we know that species from a ecosystem perspective provide different ecosystem processes and functions, while also from kind of a more forest industry perspective, also provide different sorts of products and utilities. Okay. And so in the US, the National Forest Inventory, the FIA program, is what provides a lot of the data to ultimately be used to inform these different policies that need carbon estimation. And so, as I mentioned briefly before, the forest inventory analysis data is a massive piece of information, so hundreds of thousands of locations that give information on forest resources across the US. But it was designed for doing analyses at the national level. Okay, so understanding national and regional level information on forest resources, and the kind of current statistical approaches they use are design-based approaches that require very large sample sizes to give reliable estimates. Okay, and so as soon as you try to get estimates at more finer spatial scales, so states and counties and even smaller than counties, the kind of standard approaches kind of fall apart and aren't as useful for generating estimates with reasonable amounts of uncertainty. Okay. And so this leads to the concept of small area estimation, all right, where this concept of small area estimation is kind of exactly like what it sounds. You're trying to take data that are usually designed for a larger purpose to generate estimates at smaller spatial scales, okay? And there's a ton of interest both in a kind of conservation perspective and research perspectives, as well as in the US private forest sector for improving methods for estimating forest carbon and biomass in small areas, okay? And kind of the basic concept of how this works is you're going to take the National Forest Inventory data, and you want to link that to some remote sensing data, so whether that comes from LiDAR or other types of remote sensing, use that data to improve your ability to generate estimates at small spatial scales. But you can also just tie back to that first law of geography we talked about, where if you account for the fact that locations closer together going to have more similar processes or similar values compared to those far apart, you can build that information into the models that you develop to then provide improved estimates of biomass at small scales, okay? And so really what this work, this kind of ongoing work is focused on doing is developing models. So taking that modeling framework I laid out, using it to generate species-specific estimates of biomass and carbon within very small areas across the US, okay? And the kind of application which we're working in is using these approaches to estimate county level biomass for the 20 most common tree species in the southeastern US. Okay, and just a bit of context, the southeastern US, on the left here is a map of forest ownership, and the purple hues are ones that represent private lands, okay? And so you can see here that a lot of the land in the southeastern US is privately owned, both by kind of small scale landowners as well as larger timber companies. And so if we think about the necessity to estimate forest products in small areas, it's often really applicable in this region, given just the massive amount of forest area and the products that are generated from these forests. Okay, and if we go then to look at kind of the sample sizes that we have from the forest inventory analysis data, okay, on the right here is a plot just showing how many points, how many data points we have in each county where we want to make estimates. And we can see that for many of these, we have pretty solid sample sizes. Okay, so sample sizes in the 50s and the hundreds. But where the standard models kind of fall apart is due to a lot of the data complexities that exist in species-specific forest inventory data. Okay, and so thinking about what these data look like, on the right are kind of 4 of the most common species in the southeastern US. And if you look at the distribution of biomass for these 4 species, you can see the challenges that might arise when modeling these data, particularly in the context that the standard approaches by FIA and the Forest Service use kind of classical central limit theorem-based approaches to estimating forest biomass. And what we can see is these data are highly zero-inflated. So we have locations where the species don't occur, right? So if they don't occur, then there can be no biomass there. We also see that they're positive and continuous, right? And then even after accounting for that large amount of zero inflation, we can see we have highly right-skewed data, okay? So a lot of plots with a small amount of biomass, and then a few plots with large amounts of biomass, okay? And so what we're going to do, what we're doing in this work is developing a model that kind of splits this up into 2 stages, all right? So we're calling this a 2-stage model. It's also a hurdle model, where we first do is estimate the presence or the absence of each species, and then we estimate the biomass of that species at those locations that we estimate the species to be present at, okay? All right, and this data set is pretty massive. So we have about 20 species we're working with, close to 40,000 locations, so about 780,000 data points, all right? So all those computational considerations are really important to consider when working with a data set of this size. And so kind of to quickly go through this model again, we're kind of breaking it up into two stages where we're going to estimate the presence or absence of each individual species at each location, okay? And so again, this is kind of what we have here, presence or absence of the species. We're going to estimate its distribution as a function of different abiotic variables that often come from remote sensing products, as well as account for that spatial autocorrelation in our models to improve our ability to share information across space and improve our predictions. And then because we're working with multiple species, we'll then link these again at the overall community level to improve our ability to share information across those species. Okay. And then once we estimate kind of the distribution of these species, so where they occur, what we then do is we estimate the biomass at those locations where the species occurs using a log normal hurdle model, log normal model just for this part. And then again, kind of use that sort of spatial GLM framework where we have environmental variables that impact that biomass. We have a residual correlation in our data that can improve our predictions. And then we link all of these species-specific variables at a community level, which can further improve our ability to generate estimates at very fine scales, okay? And so what we do is we fit this model with the FIA data plots that we have, and then in order to predict at individual counties and get estimates for county levels, all we do is predict at a very dense grid of points across each county, okay? So here's Wake County in North Carolina, where I live. And in order to generate an estimate of biomass for that county, we're just going to predict the presence of each species at each location. So does it occur there? Does it not occur? And then we're going to predict the biomass at each location if the species occurs there, okay? And because we do this in a Bayesian framework, we can get full uncertainty propagation and accounting for that in our estimates of uncertainty in these models. Okay, and so this is ongoing work. This approach is ongoing work, and just kind of getting some of the results coming from this now. But if we take a look at what this modeling approach can give us, it can really improve the information we can get on individual species at finer scales across the regions we're working with. Okay, so here for sweet gum, pretty common species across the Southeast, we can generate estimates of biomass along with measures of uncertainty that really allow us to quantify in a more precise way compared to existing approaches how much biomass there is for these species, where it occurs, and then ultimately use that information to inform different sorts of management questions and be used to inform different policies. Okay. And so with that, I'll just kind of wrap up by kind of acknowledging my employers and different funding sources for this work, and then the many great co-authors that have contributed to parts of this work. And happy to answer any questions if there are any. Thanks.
Oh, thank you so much. If folks can join me, let's give a virtual file. Let me see if I can. Okay. So now we have some time for question. Feel free to submit your question in Q&A box. And we'll read them out. Or you can raise your hands and I can unmute you. I'm just give folks some time to type in that question in.
Sure.
I feel like we do have one. Let me finish my that's yeah. I haven't really I'm like a beginning learner of like spatial data analysis. So I'm just curious like how you put like base model on the top of that. I don't know if that's an easy question to answer here.
So how you put a base model? So,
Yeah.
oh, a Bayesian model on.
Oh yeah, sorry, base model.
Okay. Yeah, so that's just so the Bayesian approach kind of one of the many ways you can use to analyze spatial data. That's kind of the approach that I use in my work. The reason why I use that is because it can provide you additional flexibility in trying to estimate the spatial correlation, as opposed to some of the kind of standard approaches where you have to kind of fix some of the parameters at values prior to fitting the full model. But you can do it in both different ways. So you can kind of do it in the Bayesian way, you can do it in the kind of frequentist way. In particular with some models in ecology, why the Bayesian way is useful is because then you can add in all those other sorts of bells and whistles. So you can account for the different sorts of observational biases, kind of account for correlations between species, where all of that kind of gets more complex in a lot of frequentist approaches. But you can kind of do it in different types of ways.
Thank you. So we have a question from Cole. Said, can you explain a little bit more about the nearest neighbor ordering? And more specific, yeah, go ahead.
Yeah, good question, Cole. So right, so the way to get that whole approach to work is there has to be some ordering to the spatial locations, and the way you order has been shown to not be extremely important. It's more so kind of a statistical nuance that you have to do. And so usually what you'll do is just you'll kind of find the position along either the X or the Y axis and just assign the order of each location based on where it falls on the axis. So the first location would be the one farthest to the left on the X-axis, last location farthest to the right, and so on and so forth. So the ordering, the approach you use isn't all that important. With the one caveat is that if you have extremely like clustered data, so if the data are collected kind of in a cluster design or something like that, the choice of ordering you have could actually impact the quality of the approximation. So in those cases, there's some more kind of advanced algorithms to figure out kind of a more optimal ordering to get the best result. Yeah. So even if there are closer neighbors, it doesn't have an impact on the estimates. No, I guess my short answer would be no. If you do have situations where you have like high clustering, right? If you have locations that are really, really close together, but then also really far apart, you may want to set the number of neighbors in a way that you're able to get some neighbors that are farther apart, right? So if you had a cluster of like 10, maybe you'd set the number of neighbors to 11 just to make sure you're getting some neighbors that are kind of far out of that cluster, if that makes sense.
Okay, you said I see a thanks in the chat. So I'm gonna unmute Jim because I think he had several follow-up questions. Go ahead, Jim.
Yeah, thanks. And thank you so much for the presentation. I was also curious about the NNGP and whether you'd done any comparison in terms of speed or cross-validation performance with like the SPDE method from, INLA or like Vecchia or other sort of, sparse precision construction.
Yeah, totally. Thanks, Jim. I guess I haven't in particular done a lot of those comparisons. I do think some of the there's a paper by Matt Heaton et al. In Jabes that goes through one of these kind of large scale comparisons between these different methods. All the whole suite of big N approaches, and I think the NNGP performs very comparatively to a lot of these other methods. Some of the approximation methods that are implemented without MCMC, right? Those, of course, can be used a lot faster and done a lot faster. But part of the reason I like NNGP is just that one of the things it's somewhat intuitive in terms of the concepts that it relies on, and it does kind of plug in pretty well, just kind of it being kind of a standard multivariate normal in itself, so you can kind of use it in some different ways. But it's all kind of kind of 6 1/2, right? In terms of kind of these approaches have different ways of getting at the same answer. But yeah, if you haven't seen it yet, which maybe you have, but that paper by Heaton is a really great one to look at.
Yeah, thanks so much.
So we have a question from Helen said, do you estimate all 3 species in one multivariate model, or do you do each species separately? Presumably some species will be highly correlated. How do you include this in the model?
Yeah, that's a fantastic question. So the model is a full multivariate model, so all the species are modeled together. And you're exactly right, the species are correlated with each other. And so all these models that I talked about today, these are all kind of flavors of joint species distribution models. Okay, so some models that explicitly account for correlations between those species and particularly in kind of the residuals of those species. The approach that's used in these types of models I was talking about, we use a factor modeling approach, which is kind of a way to estimate those correlations between the species, but then reduce the dimensionality of those correlations to try to make that problem a bit more computationally efficient. And that kind of factor model approach is pretty commonly used for these types of joint species models. And happy to share some more details on that if you're interested.
Yeah, we have a last question on the line if you don't mind answering.
Yeah.
We have one question from Ying Yi asked, I wonder how does the NGP approach compare to the approximation method like inducing points?
Yeah, so that kind of is similar to Jim's question in the sense that there is a whole suite of these approaches that can be used to approximate different kind of approximate that gold standard spatial Gaussian process. And there has been work that's shown that the NGP does quite well in comparison to a lot of these other approaches. But again, some of these other sort of SPDE approaches and other techniques to approximate a spatial surface oftentimes work very similar, work very well. So it kind of depends on, I guess, kind of the other aspects you want to add into the model and kind of the specific type of data you're working with to really figure out which approach works pretty well. But in kind of my experience, the NGP has worked pretty well for these approximations and yeah.
Okay, I think that's it. If anyone have a additional question, feel free to reach out. And thank you everyone for coming to the quantitative seminar today. We hope to see you next week to hear something about, sorry, let me pull that up. To hear about collective behavior in dynamic and changing oceanic ecosystem. Thank you everyone, thank you. Thank you, Dr. Loser.
Yep, and thank you.